Okay. One of the other differences between eukaryotes and prokaryotes are the machinery. There are 3 RNA polymerases in eukaryotes. They all have special jobs. Polymerase 1 axon promoters that are specific for rRNAs. RNA polymerase 3 acts on tRNAs. So very specific sets of genes. These are all functional RNAs. All the mRNAs that we're interested in, we're going to spend most of our time talking about RNA called 2, which does all the 2 mRNAs. So these are the RNAs that are going to code for proteins. We're going to focus specifically on the regulation of all 2 promoters. Alright? You don't memorize all this. You should know that the other polymerases are involved in transcribing genes for regulatory RNAs. Alright. Let's start with the 4 promoter elements. We're just going to go down that list and talk about them. There's no -10 and -35 box in eukaryotes. They have a different set of core promoter elements. All right, there is often, but not always, a kata box, very similar to what we talked about, kata box at -10 in prokaryotes, but it sits a little farther upstream generally. As well, there's another core element called DRE, which sits upstream of that. Remember, you've got to have at least 2 core elements in that promoter. Why? Let me raise your hand. Yes? Yeah, you've got to establish some kind of polarity. You've got to tell that polymerase which way to go once it binds to that promoter. And you need at least 2 core elements to do that. All right? So again, these things are similar to the -10, -35. They're analogous, but not identical. There's a couple of extra things, extra elements in eukaryotic genes. There's something called an initiator box. That sits right on the start site for transcription. Right? And then there's often other elements called downstream core elements that sit downstream of the start site. How can that work? How can you have a binding site or a core element downstream of the start site? Shouldn't that block polymerase, or I mean, how does that work? Anybody confused by that? Okay. So that polymerase, when it sits down, the polymerase is gigantic compared to this picture. So that core element can bind a protein that's just helping establish that polymerase there. Before elongation, it's going to go away. All right? So it's not going to sit there and block that polymerase. That's it. All right, questions about core promoter elements? Yeah. Well, it's going to release that initiator can't, whatever's binding that downstream core element can't sit there. It's not going to sit there because the polymerase couldn't move forward. So that polymerase is going to release from that whole complex at initiation. We'll talk about that. All right. So we don't have sigma factors in eukaryotes. The analogous set of factors are called general transcription factors, and this is a list of general transcription factors in yeast. They perform the analogous function to sigma factors. They're going to recognize these core elements, help establish that polymerase complex at the promoter before initiation. All right? There's a whole mess of them, and this is all all the information you would ever want to know and more. We're only going to focus on a couple of these different transcription factors. There's one in particular whose activities I want you to understand. And that's going to be TF2H. And we're going to talk about that in a little bit. So the concept here is eukaryotes don't have sigma factors. They have this whole set of general transcription factors. It's a class of transcription factors used at every pulse increment. All right. Let's talk a little bit about transcriptional initiation in eukaryotes. It varies a little bit from the themes that Ray talked about earlier. There's a stepwise assembly of this initiation complex. The assembly of this complex means you're going to bind all the general transcription factors in some stepwise fashion, along with Pol2, all assembled at that promoter region. We're going to go through the steps here. I don't care that you understand which ones load first. I just want to show you how this works. Okay? All right. So again, this is a cartoon. It's an example. And for a long time, people thought that there were Tata boxes at pretty much every eukaryotic gene. In fact, that's not true. One of my colleagues here, Uwe Oehler, over in the IGSP, was looking at all the promoters in Drosophila, and it's about 10% in Drosophila. Really low percentage of genes actually have Tata boxes upstream. What that means is there are other promoter elements that aren't recognizable that are probably binding these factors. Everything gets more complicated when you get to eukaryotes. It's much harder to study this stuff. It's harder to work with the organisms, and there's a lot more diversity. That's why we start with prokaryotic regulation, to give you the concepts. So this is just a cartoon. That's why I don't care that you know all of the orders here. I just want you to see how this works. So in promoters with a Tata box, a protein called Tata binding protein, along with TF2D, bind to those elements. And now you're going to get stepwise addition of a whole bunch of different other factors. All of those general transcription factors, TF2E, TF2H, and then polymerase right here, are all going to assemble at that promoter. Now you've got a giant complex of these general transcription factors and RNA polymerase at the promoter. Stepwise addition. Once you've got that complex built, you get activation of helicase activity. So TF2H is the factor that I wanted you guys to know its functions. TF2H has helicase activity. What's a helicase? Yeah, helicase unwinds that duplex. We'll talk about helicases a little more when we talk about DNA replication, but you actually have an enzyme that helps you unwind that DNA duplex. Why do you need a helicase at a promoter? Yeah, in order to transcribe, you have to be able to pull those two strands apart as that polymerase is moving, right? So you need to unwind those strands as that polymerase binds at this promoter. So TF2H is the helicase. So everything sits down, you get helicase activity, which starts to open up that duplex. And you're going to start, so you're getting all of the nucleotides feeding in there, and as you do in prokaryotes, you start to make a small RNA. But it's not making that small RNA that actually triggers the conformational change that triggers initiation. This is very important. The difference between prokaryotes and eukaryotes in this case is TF2H. So not only does it have helicase activity, it's a kinase. It's got kinase activity. What do kinases do? What kinases do? They
Phosphorylate.
phosphorylate other proteins. They put phosphate groups on other proteins. What do you think? What do you think the kinase activity of TF2H is doing? It's phosphorylating a protein. Why? It's going to regulate a protein's activity by changing its structure. TF2H. Let's go back here. On this RNA polymerase, there's an extension right here. It's called the C-terminal domain. It's a protein domain. CTD stands for C-terminal domain. TF2H is a CTD kinase. All right? So once you bind this entire complex, this complex is sitting here. It's stuck at that promoter. TF2H phosphorylates that CTD tail and swings it into a new conformation. That releases the polymerase from this whole giant complex of general transcription factors. That's how you initiate. So in order to elongate, that promoter, or that polymerase, has to escape binding from all of those transcription factors. It's got to release from that complex and move down the duplex. It does that because its CTD tail gets phosphorylated by TF2H. All right, changing conformation. Yeah.
What does that change in conformation actually do again?
Changing conformation releases RNA polymerase from being bound to that complex of general transcription factors and allows it to move down and start elongating. How is that triggered? How is that triggered in prokaryotes? Yeah, in prokaryotes, it's that abortive initiation when you finally get that small RNA that's made. When you make a long enough small RNA, that RNA is interacting with the polymerase, which changes its conformation, allowing it to release from that promoter and signal factor. Yeah. You're making a small RNA, but that's not enough to trigger the release. In this case, you're probably getting some conformational changes that triggers the phosphorylation of that CTD tail and allows that polymerase to release. There's still abortive initiation. Questions? The difference in mechanism between how transcription initiates in prokaryotes versus eukaryotes. TF2H, pretty important. Helicase and a kinase. Yes? No, actually the prokaryotes have a C-terminal domain as well, and in fact Greg showed you that C-terminal domain is interacting with those up elements in the ribosomal groups. But it's not getting phosphorylated and swung around. So it's probably some kind of adaptation during evolution. TF2H, it's a helicase, it's a kinase. Phosphorylates the C-terminal domain of RNA pol 2, triggers a conformational change. This is required for release of the polymerase. This is initiation. You have to build this complex, and in order to elongate, you've got to release from that complex. The polymerase has to release so it can move down the arc of DNA. All right, so here's the steps. We talked about them. Here's your slide. You assemble these general transcription factor complexes at the promoter. You bind the polymerase. You phosphorylate the CTD tail. Conformation release. Change that complex. Away to go. All right, so let's talk a little bit about regulation at a distance. So in prokaryotes, it's very rare to see any kind of cis-acting element greater than 500 base pairs from the promoter start site of any gene. Very rare. In higher eukaryotes, especially humans, you can see elements that are regulating a gene 50 kb upstream of the gene. Probably more than that. 100 kb, yeah. I mean, I don't even know what the limit is. Yeah, half a megabits away. This confused people for a very, very long time. And it makes trying to understand how gene regulation works in human cells very challenging. That's a whole lot of DNA to be scanning with mutations, trying to find things that regulate gene expression for one gene. So let's talk about how that works. And as well, I should point out, it's not just upstream from the gene. You can find regulatory elements that regulate transcription downstream of the gene. That's even more confusing. That you don't see in prokaryotes. Yes? So the question is there a difference between how far away they can be upstream versus downstream? You know the answer to that? Yeah, I don't know is the answer. Looking downstream gets even more difficult. Okay. So if we look at an Alberts defines this as the gene control region, which means the region where all of the elements that control transcription of a specific gene are found. It can be very large, as we just said, it can be upwards of 50 kb or bigger. In that gene control region, you can have a number of different regulatory sequences with a bunch of DNA between that doesn't seem to do anything. Right? So these cis-acting sequences are going to be binding promoter or binding some kinds of activators or repressors, some kinds of complexes. It can be very large. It can be a huge number of binding sites. Then you have the core promoter elements where the transcription factor, general transcription factors, and polymerase bind. Then you can have your gene, and then you can even have cis-acting sequences downstream that are binding proteins, activators or repressors. So we're seeing a much, much more complex set of regulatory, cis-acting regulatory elements that are binding a complex set of activators and repressors. And even the initiation complex is much more complex in eukaryotes. Now this doesn't mean every gene works this way. This means it can work this way. There are some pretty simple promoters in various eukaryotes, especially in yeast, that look a lot like the promoters that Greg was showing. They're not all super complex with elements way upstream. Just letting you know it can happen, especially in what I like to call larger eukaryotes, not yeast. All right. Questions? Yeah. The question is, do these downstream protein complexes come off after initiation? Do the upstream complexes come off too? It's not clear to me that these actually come off. No, there's an element that sits right at that. There's proteins that sit right at that start site. And there's a downstream promoter element that actually sits not downstream of the gene, but right in the untranslated region of the gene. That's got to be it. This is actually well downstream of the gene. We haven't got to translation yet, so it's hard for you to understand the untranslated genes. So there are parts of RNAs that aren't going to make protein. And that's where usually where those downstream elements are. But they're sitting right here, not downstream of the gene. Other questions? That's a good question. Let's see how that can be confusing. Yeah. General transcription factors are a class of transcription factors. Anything that binds to these regulatory elements could be called transcription factors. The difference is that general transcription factors bind to the core elements right at the promoter and are interacting in that polymerase complex, like sigma factor. So think of general transcription factors as analogous to sigma factor, and all these other things would be repressors and activators, cat binding proteins, all those kinds of things. We call them all transcription factors. General transcription factors are a very specific class of transcription factors that sit right here at those promoters. Does that make sense? Anybody else? Yes?
What?
Yeah, so this region right here, yeah, so the question is, does this region right here that's downstream of gene X, upstream of these, where these regulatory binding sites are, does that region have nothing to do with the gene yet? Potentially, yeah. Yeah, that's a great question. So the question is, could these regulatory sequences downstream of gene X actually be the regulatory sequences for gene Y that sits downstream? Absolutely. That's absolutely true. And that's exactly why things get a little tricky in large genomes. When you have these regulatory elements spaced out at large distances, there's no element, there's nothing specific about these proteins, and Greg talked about this in his last lecture. Transcription factors don't know which gene they regulate. They have no preference. So how does that work? I'm going to tell you how that works in a little bit.
No, this is promote downstream.
So there's
I think I meant that.
okay.
Okay. So how can sequences 50K be upstream or even downstream of transcription start site influence transcriptional regulation? Take a couple minutes, think about it, and let's talk about it.
Yeah, I'm gonna put these in.
Penis.
That would be mature.
That's alright.
I'm gonna put these on my iPod, and while I listen to them over winter break. Dude, I think I'm just gonna, do that. Maybe upstream.
Yeah, that's what I was thinking. It's like the fold, something that's like 100 base pairs down and actually get like.
Oh, the DNA can bend. That's
probably it, actually.
Let's just look at the next slide. Yeah. Dude, I made such stupid mistakes on the ICQ.
Dude, I got a really unhappy score I'm unhappy with.
That's not good, dude. That sucks, actually.
Really sucks. I don't even know what to do though. Like,
yeah. Well, dude, have you been to the health room at all? I feel like you should definitely go.
I should go there. I'm gonna go next time. I think they're going. Is it today?
Tomorrow.
I'm gonna go tomorrow. Ken, attending tomorrow. What time?
You're tenting tomorrow? Who's tenting?
It starts at 7. No,
You guys are both tenting?
dude, no, I'm not going to go hard on Thursday. I can go hard on Saturday or Friday. I won't drink anything.
Wait, does every tent person have to be in the tent for Thursday, Friday, and Saturday?
Yeah, Ross is gone.
All right.
I know, but that means like everyone's gonna be gone too.
Upstream, downstream,
Everyone's done.
50 kV away, doesn't matter. How does that work? Confused everybody for a really long time.
I guess and if there's a PKB upstream of the DNA bends back around where it's actually near.
All right, so if it's upstream, it can bend back around and interact with the promoter. What about downstream? How does that work? Right here. Interesting idea, yeah. So they could play some role in DNA topology. So if you don't relax those supercoils, it might be harder for that polymerase to move down the stretch of DNA. I like that. Very creative. I like that. Absolutely possible. I don't know if it happens. Anybody else?
I mean,
I like the way you're thinking. Downstream. We're talking about downstream elements. How can those influence transcription? What did he say?
No idea.
No idea. Yeah, I felt that way for a long time. Enjoy it. Ignorance is bliss.
I should be really happy then.
Exactly right. They can work the same way downstream as upstream by looping.
And there's
It doesn't matter in eukaryotes. Once you get this concept of
it's not the
looping,
same thing.
but it doesn't matter if that
Sorry,
element's
for touching
downstream
shit.
or upstream. Thanks, Greg. Looping can occur. So there are some restrictions, right? So when you're looped, if you're an element that's 50 kb away from that promoter, what's the probability that you're going to interact, that DNA is going to loop perfectly so you can interact with that initiation complex? Is it going to be higher or lower than if you were 4 kb away? What's the probability? Think about it. I have two things that are close together. Yeah, it's the probability. Well, wait a second. Which is lower? Somebody says lower. So this is the probability of something 50 kb away. Is the probability going to be higher that it's going to interact with the promoter region, or lower than the thing 4 kb away?
That's not.
Okay, good. Yes. Is that my stats guy? Okay, good. I had a stats guy. No, that's absolutely correct. There he is. So think about it. If I tie two balls together on a really, really long string, it's going to be harder for those to interact than if they're really, really close together. Okay? There is some restriction to how looping can work. Something too close together can't loop. You can't bend DNA at an angle. And I think the maximum distance is something like, or the optimal distance is something like 500 base pairs or something for looping. There are proteins that can facilitate this looping, and Greg talked a little bit about it. Mediator protein, this giant protein that's actually interacting with elements at a distance. So these elements don't actually have to get to this region. They can interact with mediator, which then interacts. There are also proteins that can interact in this cleft to help bend that DNA, to increase the probability that something at a distance can interact with that polymerase complex. And again, it doesn't matter if you're upstream or downstream, you can still make a DNA loop and interact, have those transcription factors that are bound at a distance interact with this initiation complex. All right? And activators, as Greg pointed out, I'm going to say it again because it's important. How do activators work? They act to stabilize this initiation complex. In any way that they can bring in either general transcription factors or the polymerase and stabilize that initiation complex, it'll be an activator. A repressor is any way you can think of that you destabilize that complex. Activators and repressors. Yeah.
What did you say?
Yeah, so a great question. So the probability of interacting with that promoter region is higher if you're closer. What's the point of having them far away? I have no idea. Because it works. It worked in evolution. So remember, evolution doesn't always work to optimize any system. It just works for that organism at any given time. Yeah. Well, that would be one reason. So one reason it might be well about, at a great distance from that gene is that it's involved in regulating other expression from other genes. That can definitely happen. So this sequence that's at a distance up here doesn't necessarily know it's supposed to act with that promoter. It can interact with any other promoter that's in range. All right? No specificity for these transcription factors. There's specificity for binding to a DNA element, but they have no idea which promoter they're supposed to interact with. It's a really important point. Is anybody confused by this? Okay. Asked to repeat. So, transcription factors, this transcription factor bound to that element, it's specific for binding that element, but it has no idea what promoter it's supposed to interact with. It doesn't care. There's no specificity. And Greg showed you that slide last time where he had the GAL4 transcription factor, swapped out the activating unit for GAL4, and it still could trigger as long as you had the GAL4 binding site. Transcription factors are blind to who they interact with, except for that sequence. They're specific for the sequence, not for a promoter. Questions? Okay. So why have regulatory sequences downstream of the gene? Kind of already talked about this, so I'm not going to bust you guys up. Because you can, is the answer. Because you can. Regulatory elements associated with transcription factors don't care which promoter they act on. So regulation at a distance in a dense genome could mean the transcription of multiple genes could be influenced. We talked about this. Sometimes this is by design. Why would you want one transcription? Let's talk about that. Why would you want one transcriptional element regulating several different genes? Yes. You might want to produce several things all at the same time, given some signal that comes in and says, hey, turn this stuff on. How do prokaryotes do that? Anybody? Operons. They do it with operons. Eukaryotic operons are very rare. And I'll tell you why that is when we get to translation. Eukaryotic operons are not rare because transcription is different. It's because translation in eukaryotes is different. So I'll tell you why that is later. Yeah. Yeah, so question is if some upstream sequence is regulating a couple of different promoters, could it activate one and repress another? Yeah, it could. Activators can act like repressors at times. Greg talked about that. This is why we're teaching you the rules. I could give you 500 examples of things and still not cover everything. If it doesn't break the rules, it probably happens in some gene. This is why we don't give you 4,000 examples of genes and how they're regulated. I'd much rather teach you the rules. Yes? Yeah, so you don't normally, and it's not an always thing, you don't normally see regulation at a distance in high-density genomes, but in the case where you wanted to regulate several genes at once, Yeah, it would make sense to do that. Yes. Now, I did say that general transcription factors are not specific for a promoter. I said that the upstream transcription factors are not specific for any given promoter. They can loop and bind to any promoter.
They're saying there's always exceptions.
General transcription factors bind specifically to those core elements. No, yeah, general transcription, it's just like sigma factors, right? So sigma factors are going to bind specifically to -35 in kata, and some are better at binding various consensus sequences than others.
That's why I'm gonna watch
Same deal.
lectures. It's okay.
Anybody else? All right. So, is there a way that cells can prevent a regulatory sequence that's well up from a gene from regulating a neighboring gene? All right? Because this is an issue for sure in high-density genomes, or if you have genes next door that you don't want to be regulated by a specific cis-acting sequence. You don't want looping to happen. How do cells take care of that? They have a mechanism to do this. There are actually two mechanisms. One's called an insulator element, and the other is called a barrier sequence. The best way to think about, the way I think about these two kinds of sequences and elements, and these are just names, sequence and element are interchangeable. The best way to think about it is if I have an enhancer. What's an enhancer? Hello. It's a cis-acting sequence that binds an activating transcription factor. Sometimes they're repressing, but people have called them enhancers over the years. So if I have an enhancer which is going to bind some transcription factor, and it's specific for gene B, if I have a neighboring gene A, how do I block gene A from getting transcribed or acted on by this enhancer element? I can insert an insulator element. The way I think about this, and I can't tell you what the exact the mechanism is, it's going to bind some protein, and I think of that like a large fence. Think about putting up a fence here, so that I can't get looping of this enhancer to the promoter of gene A, but I can get looping of this enhancer to gene B. Now that element could act by bending the DNA in such a way that this has an easier time of interacting with that than that. I don't know for sure. Do you know, Greg? Okay. So for the purposes of this course, you're going to think about the insulator element as a giant fence. You put it between an enhancer upstream and a gene, that enhancer is going to work on the gene this way instead of that way. All right? It's got to sit between. Yeah. No. Great question. Does it promote the transcription of gene B, or does it just block that enhancer from acting on gene A? The best way to think about it is that it just blocks that enhancer from acting on gene A. Question about insulator elements. Okay. The other sequence, called a barrier sequence, works a little bit differently. And we're about to talk about the influence of chromatin on gene expression. Barrier sequences prevent the spread of heterochromatin into regions where genes are being expressed. So we'll talk a little bit about heterochromatin, and you initiate this. We talked about heterochromatin before. You initiate heterochromatin in various regions, and then it spreads from one site. This barrier sequence prevents the spread of heterochromatin, which is going to shut off gene expression in neighboring areas. All right, so these two elements are important. People started to learn about these or become more interested in them when we started making transgenic animals. So when people make transgenic animals, they're putting in a gene with some regulatory sequences, and you stick it in the cell, you don't know where it's going to go. And the problem was, if you had a bunch of regulatory elements stuck to it that you could manipulate, if you wanted to manipulate the expression of this gene in the cell, what happens is if it goes into an area where there are a bunch of other enhancers or regulatory elements, then you've lost control of that gene. So what people do is they insert these insulator and barrier elements upstream and downstream of their genes to make sure that they can regulate it with the sequences that they put in there. All right? Okay. So this is how a cell can deal with regulation and distance and prevent the inappropriate regulation of some other gene. All right, let's talk about chromatin. We talked about how nucleosomes can influence transcription initiation. We'll be a little more specific today about it. We're going to talk about chromatin remodeling complexes, which can basically unravel the DNA from these nucleosomes and expose core promoter sequences. It's a process called remodeling. Buzzword you'll hear all the time, chromatin remodeling. We're going to think about it in terms of transcriptional regulation as unraveling the DNA, but remodeling can also mean, is re-raveling the word? Re-raveling the DNA and putting it in a closed conformation. So this is a two-way reaction. This closing up of DNA is the essence of constructing heterochromatin. You'll also hear it called gene silencing. I'm not going to talk too much about silencing. All right, so remember, prokaryotes don't have to deal with histones. The DNA, for our purposes, is reasonably naked, and not true in eukaryotes. So how does this work? So euchromatin, as Greg pointed out, genes that are going to be transcribed are in this kind of beads on a string orientation. AC here stands for acetyl groups. So open conformations of DNA, like beads on a string. Remember, we talked about the fact that histone tails get modified like crazy. One of the modifications that they get is an acetylation. And the acetylation of these histone tails actually opens up the chromatin. P here stands for phosphorylation, ME for methylation. Just to show you that open conformations of DNA don't have acetyl groups at every histone tail. This is the histone code we talked about last time. Why would acetylation of a histone tail open up that conformation? Why would that take you from this conformation to that conformation? How does that loosen up the interaction between the DNA and the histone? What do you guys know about histone tails? Besides the fact they get modified like crazy, yeah. They're very positively charged. So you put a cell group on them. Where's that positive charge going to interact with? Sugar phosphate backbone. Now, let's not forget our biochemistry. All right, so putting a cell group on there actually allows that, changes the charge distribution on that tail. That's going to make it not interact with that sugar phosphate backbone as easily. There's a number of other reasons as well, and we're not going to get into them. So heterochromatin tends to be heavily methylated rather than acetylated. This isn't an all or nothing thing. This is a gross generalization, and it's good enough for today. All right, so chromatin wrapped up in this configuration, how our transcription factor is going to recognize a promoter that's buried in that wall of histones, this 30 nanometer filament. you're going to have a really hard time doing it. Why heterochromatin tends to be silenced. All right. So let's talk a little bit about chromatin remodeling. There are a number of ways that chromatin remodeling can occur. Gene activator proteins can actually, not just about, there's another mechanism by which transcriptional activators can activate transcription in eukaryotes. In prokaryotes, it's all about stabilizing that initiation complex. In eukaryotes, it can also be the recruitment of chromatin remodeling complexes that open up the conformation of DNA. There's another difference between prokaryotes and eukaryotes. Activators, not recruiting polymerase, not stabilizing the initiation complex, but actually recruiting chromatin remodeling complexes. I'm just giving you guys the answers right now. I want you to know the difference between prokaryotes and eukaryotes. So how does that work? Well, you can have a gene activator protein that binds to a specific sequence. There's the TATA sequence all buried up in those nucleosomes. TATA binding factor is not going to be able to get there. This gene activator protein, which here is shown binding on the sequence outside of that nucleosome, so it's getting access to it, brings in a chromatin remodeling complex that then opens up the nucleosome region near that TATA box. There's a number of different kinds of remodeling complexes. I don't care that you know them. All right, so there are histone chaperones that do various things. They can either modify the histones there, or they can strip them off. Very often you have nucleosomes stripped off the DNA, in this case, to open it up so TATA binding factor can recognize that binding site and initiation can occur. So in this example, a gene activator protein isn't interacting with the promoter region or polymerase or anything. It's just bringing in this remodeling complex to strip off those histones so all those other proteins can bind. Questions? That's all I want you to take away from this slide. Yeah? Yeah, the question is, histones, when the histones are replaced, are the exact same histones? It's unlikely. As soon as they come off, they're going to float away. So there's nothing latched onto that DNA, pulls off the histones and holds them there and then relatches, right? They're going to float away. Other questions? Yeah. Right, so a histone modifying enzyme in this case, something that's going to open up chromatin is going to be an acetylase. That enzyme is called a histone acetyltransferase, or HAT. What do you suppose the enzyme is going to do that converts this converts the chromatin back to heterochromatin? It's a deacetylase, histone deacetylase, H-dash, right? So these reactions are reversible. Okay, so you can strip the histones off, or you can bring in a modifying enzyme to open up that 30 nanometer filament. New kind of activator. All right, so chromatin remodeling complexes can also be repressive. So the same goes for repressors in eukaryotes. They're not always acting to destabilize this initiation complex. In some cases, they're acting to recruit chromatin remodeling complexes that take open conformations of chromatin and convert them to closed heterochromatin. All right, so how does that work? Well, here we have a repressor in red. We already have an open region, that promoter region, and then that repressor can recruit a remodeling complex that actually turns this back into a closed region. Okay? Now we've added another mechanism for repressors. Yeah? So say it one more time.
So why would it
Right, it could be open because it was previously activated and you want to shut that promoter off. Yeah. Well, yeah, so the question is, why do you need a barrier if this is an active process? There are several levels of heterochromatin formation, and we're not talking about all of it. So once you form this kind of 30 nanometer filament, this kind of structure, you can stack other proteins on top of that and make it even more difficult to unwind. Okay, so in this case, as we said, histone acetylases put acetyl groups onto these histone tails, which opens up that conformation. Histone deacetylases are going to strip them off, which can reclose that conformation. As well, you can recruit methyltransferases. Methylated histone tails tend to be associated with heterochromatin. And in this case, in answer to your question, when you methylate, those methyl groups actually bind other proteins that help really close up that DNA and make it inaccessible. These tend to be called silencing proteins, SIR proteins, sirtuins. Okay. All right. So for a long time, people thought about chromatin remodeling as something that happened in which at times when you're converting heterochromatin to euchromatin. They thought it was going on in large stretches of DNA, and as it turns out, it looks like remodeling occurs at promoters each time they're activated or repressed. So even very highly expressed promoters that we study in yeast, a promoter called the Gal promoter, it looks like each time initiation occurs, there's some remodeling going on. So these chromatin remodeling complexes are very, very important, not just for controlling heterochromatin versus euchromatin, but opening up that chromatin in the very last little bit and exposing those promoter regions so that they can be bound by general transcription factors. Yes? Yeah, it's going to happen at a pretty rapid rate. Energy wasted, you know, I don't know how energetic this process is, but again, it's energy regulation in eukaryotes, especially multicellular, is probably a little less important than a prokaryote or single cell organism that's trying to replicate as fast as it can. All right. So this seems to happen at promoters pretty much all the time, every time they're initiating transcription. We're getting remodeling events. How does it work? Well, again, really, really complicated slide. So let's simplify this. What I want you to remember, as I told you, these histone tails are getting modified like crazy. This is a stepwise activation of a gene. An activator is bringing in a chromatin remodeling complex that puts in a cell group here. There's a Tata box. Once you get that cell group there, another modifying enzyme, kinase, comes in. And that kinase then makes another modification, allowing TF2B to now bind. TF2B actually has binding sites, not just for the specific sequences on the DNA, but for the modifications on the on the histone tails. So some of these transcription factors are not just binding specifically to DNA sequences, but specific modifications on histone tails. This is a general transcription factor, right? So you're finding this at every promoter. It's interacting with these histone tails. All right, and that's going to lead to the assembly of this whole complex. So if you want to think about what's happening at a promoter in eukaryotes, it looks something like this. First, you're going to have some activator that's going to bring in a chromatin remodeling complex. You're going to get histone modifications, binding of other activator proteins. Then you're opening up that promoter so general transcription factors can bind. They're going to interact with all of the other regulatory proteins. Then you're going to assemble this initiation complex, and then you get initiation. There's a whole lot of stuff going on. You guys are going to remember one thing. This is a good slide to study from. Good slide to study from.
That's weird.
Question. This is just a list. I want you guys to understand the complex, or the concepts here. Yeah. Are there any of these things that can't be regulated? Gosh, any type of protein function can be regulated by modification. The only thing that can't be regulated are the DNA sequences that are sitting near the genes. These are the directions. They don't change. They change over evolutionary time, but the DNA sequence stays the same. It's the proteins that are doing everything else. Yes?
You said that deacetylation and methylation both like condense the chromatin. Is there a difference between them?
Yeah, so the question is that deacetylases and methylases are both enforcing more closed chromatin conformations. Is there a difference between them? Yeah, they're carried out by two different enzymes.
Okay, they don't. They just both have the same function.
They both have they don't have the same function. They have the same outcome. The function of the deacetylase is to strip the acetyl groups off. The methylase is actually going to put methyl groups on that allow other proteins to bind to put it into a more closed conformation. So the deacetylase is taking it from a very open conformation to a somewhat closed conformation. The methylase is taking it from a more closed conformation to an even more closed conformation by bringing in these silencing proteins.
Okay.
Does that make sense?
Yeah.
The outcome is kind of the same, but it's two different levels of chromatin packing. Is one way to think about it. Does that help?
Yeah.
Okay. So transcription initiation is probabilistic. All right, we're not going to think about transcription initiation anymore as an on-off switch. I mean, this slide's ridiculous, right? All of these things are happening each time we turn on a promoter. They're all regulated. All of these steps can be regulated to some degree. We now have a whole bunch of different sequences that are binding a whole bunch of different regulatory proteins. Transcription factors can be 50 kb upstream, and are they going to interact or not? This is not an on-off switch. Your ability to initiate is going to be a function of which and when of all these different steps occur. And transcription initiation can be influenced by any of them. All right? So why do cells do that? Why do eukaryotes do this? Promoters that are integrating all of these different inputs, all these different steps that can be regulated, allows them to really fine-tune the rate of transcription initiation. A lot of fine-tuning going on. Eukaryotic genes can respond to a huge number of inputs, signals from outside the cell. So when we think about it, it's not like this activating complex binds and so we turn on at a high level. That's not how it works. This gene down here is going to be its initiation, the amount of initiation we see from this gene is going to be some function of how many of these strongly activating complexes do we have, how many inhibiting complexes do we have, how well this bend occurs. We can have weakly activating protein complexes or transcription factors. So initiation is going to depend on which of these are bound at any particular time, and how well this DNA is bending. It can be affected by topology, supercoiling, all kinds of things. Alright? What I want to leave you with, this slide, is that it's not an on-off switch. This is not a light switch. The amount of transcription, what's the difference between a strongly expressed gene and a weakly expressed gene? Yes. Yeah, it's the interaction, but what does it mean in a absolute sense? Yeah. It's the number of mRNAs you make, and what's the rate limiting step in transcription? It's initiation. It's how many times you initiate. So a very strongly expressed gene is one in which you're going to have a whole bunch of polymerases in a train going down that DNA sequence with all the RNAs coming out of it, just like Greg showed. A weakly expressed gene is going to initiate far less often. So the number of polymerases that initiate and are transcribing a gene are going to be a function of all of the different elements, whether they're chromatin remodeling or transcription factors that are binding the cis-actin sequences. It's going to be a function of all of those things that are attached. This allows the cells to fine-tune expression. Because all of these transcription factors in eukaryotes, for a large number of them, are going to be responding to signals. So Greg talked about signals in prokaryotes, like do I have tryptophan or not? Or do I have glucose or not? Or do I have lactose or not? There are different kinds of signals for eukaryotes, and we could spend the next 3 weeks talking about all of them. And we won't. Unless you guys want to. That's it. See you guys on Friday.
James has.
Yeah, I gotta ask the TA something. He said test corrections. They said test corrections would be there Monday. Nothing's been addressed. Pisses me off. There we go.
Pisses me off. I study, just pisses me off.
What?
I'm so bad at this class, it's like nothing I can do.
You should go to the help room, you weirdo. That's the help room's made for exactly.
Why? Why would I want to get that? I give class.
Whoa.